May peace be on all of you. Myself, Dr. Majid Khan, Associate Professor, Loknethe Dr. J.D. Pawar College of Pharmacy, Kalwan, Nasik. I welcome all of you in this chapter Antihistamine. So in this video, that is in lecture 3, we will cover second generation antihistamine, mast cell stabilizer, H2 blocker and proton pump inhibitor. Learning goal. The student learning goals: second generation antihistamine and mast cell stabilizer, and that their mechanism and drugs. Then development of H2 antagonist SAR and receptor interaction of H2 antagonist, proton pump inhibitor and their development and mechanism of action. So now let us start second generation antihistamine. So as we seen in previous lecture, in classification there are two types: first generation and second generation. First generation, that is older drug which having the side effect sedation, and second generation is newer drug. So in case of second generation antihistamine, initial design efforts toward new or second generation antihistamine focused on developing agent with lower sedation potential and reduced binding affinity for non-target protein, including muscarinic, adrenergic and serotonergic receptor. These efforts led to introduction of estimazole and terfenadine, derivative of first generation agent, with substantially larger N-tertiary amine substituent. So the first generation agent having the some side effect, like it causes sedation and there is a lack of specificity regarding the receptor. So first generation agent, apart from histamine blocking, it also block muscarinic, adrenergic and serotonergic receptor. But in second generation is developed to reduce the side effect, that is sedation, and to reduce the effect of these drug on the other receptors. So in case of second generation agent, estimazole and terfenadine approved to be non-sedating and targeted receptor selective, but extensive clinical use uncovered serious cardiovascular toxicities. So this second generation agent are non-sedating and target receptor selectivity, but it having another side effect, that is cardiac arrhythmia due to blockade of potassium channel in cardiac tissue, and such effect observed commonly when this drug administered with other drug that inhibit cytochrome metabolism, such as ketoconazole, itraconazole, erythromycin and clarithromycin. So this second generation drug is developed to reduce the side effect of first generation, that is sedation, and target of other receptor like muscarinic, adrenergic, etc. So these second generation are non-sedative and target specific receptor, but apart it also have side effect, that is cardiac toxicity or cardiac arrhythmia, which is due to the blockade of potassium channel, especially these drug when administered with other drug like ketoconazole, itraconazole, erythromycin, etc. Further, the drug design effort result in discovery of new second generation agent which retain non-sedating receptor selectivity property but lack cardiotoxicity of estimazole and terfenadine. And these include acrivastatin, cetirizine, levocetirizine, fexofenadine, desloratadine, etc. So initially, in case of second generation, estimazole and terfenadine is discovered as a second generation agent. So these agent having less sedative effect and it act on a specific target receptor, but these drugs have side effect cardiotoxicity. Then after this, a new drug is also discovered, that is second generation new drug, which not have this cardiotoxicity effect, and it include acrivastatin, cetirizine, levocetirizine, fexofenadine, etc. So in addition to their H1 receptor selectivity, these second generation agents do not accumulate in the CNS because their hydrophilicity and high affinity for P-glycoprotein efflux pump to cell associated with blood-brain barrier. So these are the small classification of second generation agents. In that there are three classes: piperidine derivative, example terfenadine, fexofenadine, estimazole. Second one is piperazine derivative, example cetirizine. And third one is pyrrolidine derivative, in that acrivastatin. So it is the classification of non-sedating drugs. So piperidine derivative, terfenadine, fexofenadine, estimazole. Then piperazine derivative, cetirizine, pyrrolidine, acrivastatin. So these are the structure of this drug, estimazole. Then terfenadine, fexofenadine. So here the difference only is about the R. This one R, when terfenadine R equal to CH3, fexofenadine it is carboxylic acid. So it is a small classification of second generation. Again example is cetirizine, second generation, acrivastatin, it is a second generation. So either cetirizine is indicated for temporary relief of runny nose, sneezing, itching nose or throat, or itchy watery eyes caused by hay fever, it is allergic fever, or other upper respiratory allergies. So it is about the second generation. So in exam, short note on second generation or called as non-sedating antihistamine may ask. So first generation having the side effect, that is sedation, and other receptor also affected by first generation. So to overcome this, second generation is developed. So first second generation is developed is estimazole and terfenadine. So these drugs having less sedative effect and it having the specific target, act on a specific target but not affect the other receptors. But these estimazole and terfenadine having the side effect cardiotoxicity. Then new drug is developed, that is cetirizine, levocetirizine, acrivastatin. So this drug does not have cardiac toxicity, but it also have less sedation effect and receptor selectivity effect. So it is there are three types of classes of second generation, that is piperidine derivative, piperazine derivative, and pyrrolidine derivative. So it is about the second generation antihistamine. Now next is, that is mast cell stabilizer, or called as inhibition of histamine release. So the discovery of the bronchodilating activity of natural product choline obtained from the plant Amiresmoga led to development of compound that stabilize mast cell and inhibit the release of histamine and other mediator of inflammation. The first therapeutically significant member of this class was cromolyn sodium. Further research targeting more effective agent resulted in the introduction of nedocromyl, followed by more recently pemirolast and iodoxantid. So these are some mast cell stabilizer. Generally the mast cell stabilizer inhibit activation of and mediator release from various inflammatory cell type associated with allergy and asthma, including eosinophil, neutrophil, macrophages, mast cell, monocyte, and platelet. In addition to histamine, these drugs inhibit the release of other mediators like leukotrienes, prostaglandin, etc. So As name indicate, mast cell stabilizer. So these drugs only inhibit the release of mediators, which may be histamine, which may be leukotrienes. Therefore, it prevents the action of histamine. Therefore, it is called as mast cell stabilizer. The example of this category is chromyl sodium. So chromonyl sodium, it is a structure of chromonyl sodium, that is mast cell stabilizer. Second example, that is nadochromyl. So it is a structure of nadochromyl. So it is medication considered as a mast cell stabilizer, which act to prevent wheezing, shortness of breath, other breathing problem caused by asthma. It is administered by an inhaler under the brand name Tilade and as an eye drop under the brand name Allocril. So these are the mast cell stabilizer. Generally these drugs are used in asthma condition. So example of mast cell stabilizer is chromyl sodium, and then this one nadochromyl. These are the mast cell stabilizers. Then next class is, which is important one, H2 blocker or H2 antagonist. So in exam many times this question asked about the short note about H2 blocker or H2 antagonist. So H2 blocker. So here the acid secretory unit of the gastric and mucosa is the parietal cell. This parietal cell contains hydrogen potassium ATPase or hydrogen ion pump that secrete proton in exchange for the uptake of potassium ion. Secretion of acid by gastric parietal cell is stimulated by various mediator receptor located on the basolateral membrane, including histamine agonism of H2 receptor, gastric activity at G receptor, acetylcholine at muscarinic receptor. Gastric and muscarinic receptor mediate acid secretion through calcium dependent process, while H2 receptor is coupled with adenyl cyclase pathway. So regarding mechanism of H2 blocker, so these drugs you mainly use as an anti-ulcer or to reduce the acidity. So as we know, in stomach there is a parietal cell. So it is the parietal cell. In parietal cell there are various receptors are located. For example, histamine receptor, especially H2 receptor. Then there is a gastrin receptor. It is called a G receptor. This one is G receptor for gastrin. Similarly, muscarinic receptor for acetylcholine. So these are the receptors present in the parietal cell. So here the acid secretion is controlled. So it is the pump, that is hydrogen potassium pump. So here the hydrogen is released as an acid through this pump, and when this secretion of acid is controlled by various receptor mediated process. For example, if histamine bind to their receptor, that is H2 receptor, there is an increase the concentration of cyclic AMP by increasing the activity of adenyl cyclase, which causes release of H plus ion and reabsorption of potassium ion. Similarly, gastrin and acetylcholine, the action of this gastrin and acetylcholine is depend upon the calcium. So gastrin bind to their receptor G, acetylcholine bind to their receptor muscarinic receptor, and that increase the calcium and also stimulate this same receptor, that is secretion of H plus ion. So here the adenyl cyclase pathway, and therefore acid secretion is inhibited by intracellular prostaglandin of E series, that is prostaglandin E. This prostaglandin also stimulate other GIT epithelial cell to secrete mucus and bicarbonate and enhance blood flow through the gastric tissue. All of these prostaglandin mediated action have acid neutralizing and gastric tissue protective property called as cytoprotection. When there is a hypersecretion of gastric acid or breakdown of mucosal cell defense, including bacterial infection, gastric tissue, and contiguous structure like esophagus, intestine, etc., may become compromised and ulcerated. So it is a normal process how acid is secreted in stomach. So through this various receptor, H2 receptor for histamine, G receptor for gastrin, muscarine receptor for acetylcholine. So acetylcholine causes the secretion of acid through the cyclic AMP as a secondary messenger and through the stimulation of this pump, that is potassium hydrogen pump, and remaining gastric acid, gastrin and acetylcholine causes the acid secretion through the calcium dependent process. So especially this pathway, that is cyclic AMP pathway, is inhibited by prostaglandin E. Prostaglandin E is a chemical in our body. So this prostaglandin have cytoprotective effect. Cytoprotective effect means decrease the H plus ion and increase the secretion of bicarbonate, it neutralize the acid. So prostaglandin inhibit this effect and it produces the cytoprotective effect. So here regarding this topic, that is histamine increase the level of cyclic AMP and causes the release of H plus ion, means acid. So our drug that is called as H2 blocker, as name indicate, these are the drug or agent which simply block this H2 receptor. So if H2 receptor is blocked, then the level of cyclic AMP decreases and there is a less secretion of H plus ion. So in short, it decreases the acidity and therefore it acts as a anti-ulcer. So H2 blockers simply block H2 receptor and produce anti-secretory effect. So it is the mechanism of action of H2 blocker. Now SAR of H2 blocker, it is important one. So in case of SAR, or it is also called as development of cimetidine. Cimetidine is the first H2 antagonist. So SAR or called as development of cimetidine. So cimetidine developed by classical structural activity study beginning with endogenous agonist histamine. So it is a histamine. So it is a agonist on H1 and H2 receptors. So histamine act as a agonist at H1 and H2 receptor. So histamine act on both H1 and H2 equally. So first point, methylation of fifth position of histamine selective toward H2 agonist. So histamine, start from the histamine. It is the basic chemical in our body, histamine. It acts on both receptor H1 and H2 equally. But if we do methylation on fifth position, so it is a fifth position of histamine. So here is a methylation on fifth position. It gives 5-methyl histamine. It having the selectivity toward the H2 receptor is more as compared to H1. So due to this methylation on fifth position of histamine, it produces the 5-methyl histamine which having more activity on H2 receptor as compared to H1 receptor. Second point, the gonadino analog of histamine was then found to possess weak antagonist activity to acid secretory action of histamine. So where there is a gonadino analog of histamine, so it is a structure of histamine. So here one hydrogen is replaced by gonadino group. So it gives N-guanyl histamine. So this N-guanyl histamine is a weak antagonist. Remember here histamine is an agonist, but if we do gonadin group attach here, then the activities changes from agonist to weak antagonist. So this 5-guanyl histamine is a weak antagonist on H1 and H2 receptor. Next point, increasing the length of side chain from 2 to 4 carbon coupled with replacement of strongly basic guanine in a group by methyl thiourea leads to development of buramamide, the first fully H2 receptor antagonist but with low potency. So here there are two changes is made in histamine. There is change in the length. So here in case of histamine there are 2 carbon, but here there are 4 carbon. And second change, that is guanine group is replaced by methyl thiourea. So it is a methyl thiourea group. So it gives a new drug called as buramamide. So it is a buramamide. It is the first fully H2 antagonist but with low potency. So here two changes should be there. Number of carbon increases in side chain from 2 to 4 carbon, and guanine group is replaced with methyl thiourea. This one is methyl thiourea group. So it is a full antagonist with low potency. The next point, introduction. So here this buramamide having low potency. The low potency of buramamide was postulated to be related to its electron releasing side chain, which favor N-pros tautomer in histamine, which predominantly prevent higher affinity to N-tale tautomer to the receptor. So this drug having the low potency, and this low potency is due to the electron releasing group. So it is an electron releasing group, due to which this drug show low potency. But introduction or insertion of electron withdrawing thioether side chain in place of methylene group favor N-tale tautomer, and introduction of 5-methyl group favor H2 selectivity, and it gives the new compound metiamide, which having H2 blocker activity with high potency, and oral bioavailability is also there in case of metiamide. So it is a structure of metiamide. So here there is a 5-methyl group is also there, which increase the activity of this drug towards the H2 receptor more, and there is a again thioether is there. So here there is a thioether. So here the sulfur is added, thioether. So due to which it is having more activity and favor the H2 receptor activity. Then next point, replacement of thiourea moiety of metiamide with cyano function produce cimetidine. So here in case of metiamide there is a thiourea, but here thiourea is replaced by cyanoimino function. It gives the drug that is cimetidine. So cimetidine, it is the first drug coming to the market and used as a H2 blocker. Cimetidine promote healing of duodenal ulcer. It is short acting and required frequent dosing schedule. It possesses anti-androgenic activity which leads to gynecomastia to cause confusion state, and this effect is due to imidazole ring. So cimetidine is obtained. It having good anti-ulcer activity, but it also has some side effect called as gynecomastia. Gynecomastia means appearance of female character in male. That is called as gynecomastia, and this side effect is due to this imidazole ring. So it is the first drug, that is cimetidine, which used as a H2 blocker, but it having the side effect gynecomastia, which is due to this imidazole ring. Next point, replacement of imidazole ring with furon, example ranitidine, or with thiazole, example famotidine, nizatidine, with basing ring substituent, example guanidine or dimethyl aminomythyl, not only enhance both potency and selectivity but also reduce the side effect. So in case of cimetidine, the side effect gynecomastia is due to this ring, that is imidazole ring. So in case of new drug, this imidazole is replaced by furon, it may be replaced by thiazole, then it gives a new drug, that is a ranitidine, famotidine, nizatidine. So these are the drug where the imidazole is replaced by, so it is a cimetidine structure, but here there is a thiazole, here also there is a thiazole, then here there is a furon, that is a ranitidine, nizatidine, famotidine. So these are the new H2 blocker. So it is about the H2 antagonist or H2 receptor and antagonist. So here in case of H2 antagonist, we were seeing mechanism of action and their SAR. Now the next is, that is last class, proton pump inhibitor called as PPI. The final regarding mechanism of action, the final step in acid secretion is parietal cell is the extrusion or pumping of proton into the lumen of stomach by membrane hydrogen potassium ATPase pump. Thus inhibition of this proton pump act beyond the site of action of secondary messenger, that is calcium and cyclic MP, and is independent of the action of histamine, gastrin and acetylcholine. Thus acid pump inhibitor block the both basal and stimulated acid secretion. So proton pump inhibitor, as name indicate, these are the drugs which inhibit the proton pump. So as we seen in previous category, there is a parietal cell, there is a receptors, and there is a pump. Here is a pump. So proton pump inhibitor act on this pump. It does not have any effect on this process. So proton pump inhibitor not have effect on H2 receptor. It does not have effect on gastrin and muscarinic receptor. They directly inhibit this pump. Therefore it is called as proton pump inhibitor. So this proton pump having function, normal function of this pump is to secrete the H plus ion and to reabsorb the potassium. So when this drug inhibits this pump, then there is a less secretion of H plus ion. Therefore these drugs have anti-ulcer or use in treatment of acidity. So it is the mechanism of action. These drugs simply inhibit this pump, hydrogen proton potassium pump. Therefore there is a less secretion of acid in the stomach. So it is the mechanism of action. So here if we see chemically how they act, so these drugs inhibit the pump, and generally these drugs are the derivative of pyrimidine methyl benzimidazole. So it is the general structure of this category, proton pump inhibitor. It is a pyridyl, so it is a pyridyl ring. Methyl benzimidazole, so it is a methyl benzimidazole. So these are the class general structure of this type of drug. Now how this drug act? So the benzimidazole proton pump inhibitor are the prodrug that are rapidly converted into sulfonamide. It is a sulfonamide intermediate in the highly acidic environment of gastric parietal cell. The weakly basic benzimidazole derivative proton pump inhibitor accumulate in this acidic environment. The benzimidazole proton pump inhibitor are then chemically converted by acid to sulfonamide. It is a sulfonamide, not sulfonamide. So note the point, it is a sulfonamide intermediate that inhibit the proton pump via covalent interaction with sulfhydryl group with cysteine residue of the hydrogen potassium ATPase pump as shown in this reaction. So these drugs, that is proton pump inhibitor, act as a prodrug. These drugs inhibit the proton potassium pump. So it is the reaction how these drugs first activate into sulfonamide. It is like a prodrug. So it is a general structure of this drug, that is proton pump inhibitor. So first step here, there is a protonation. So here there is a protonation taken place like this. So it is a protonation. Then in step 2, there is a attack of lone pair of nitrogen. So in step 2, this lone pair attack to here carbon, then this double bond is break and this charge is neutralized. So here there is a formation of bond or 5-member ring. Then third step, removal of hydrogen. This bond shifted here and this bond is break and it become O minus, then hydrogen shifted, it become OH like this. Then again there is a lone pair of nitrogen attack to this sulfur, a removal of water molecule, OH from here, hydrogen from here. Then there is a formation of this called as sulfonamide. So this sulfonamide is an active form of proton pump inhibitor. Then this sulfonamide react with the sulfhydryl group present in cysteine of hydrogen potassium pump. So potassium hydrogen pump consists of amino acid cysteine, and that cysteine having the sulfhydryl group SH. So this sulfonamide react with this SH group of proton pump by this way, and there is a formation of complex. So it is a formation of covalent bond complex of proton pump inhibitor and proton pump. So by this reaction they inhibit the proton pump. So again PPI, that is proton pump inhibitor, is a benzimidazole derivative. It acts as a prodrug. First these drugs activate into their active form called as sulfonamide. So here there is an activation reaction of proton pump inhibitor. So in that first step is a protonation of this nitrogen. Then this lone pair attack to this carbon, then charge is neutralized on this carbon. Then there is a hydrogen is removed, this bond shifted here, it become double bond, this bond is break, this carbon will become O minus, hydrogen transfer here, it become OH. Then again lone pair of this nitrogen attack to this, there is a formation of ring and removal of water molecule. So this the intermediate is form is called as sulfonamide. Then this sulfonamide react with the sulfhydryl group of cysteine of hydrogen potassium ATPase pump, and it forms the covalent complex between proton pump and proton pump inhibitors drug. So by this way it acts as a proton pump inhibitor. All of these PPI have been shown to react with cysteine 813 of pump, but some bind to additional pump sulfhydryl residue as well. So the main target of this drug is cysteine of the position 813, but it also inhibits the other cysteine molecule like omeprazole, isoprene also react with cysteine 892. So cysteine 813 is the main target, but omeprazole, isoprene also inhibits cysteine 892, pantoprazole inhibits cysteine 822, lansoprazole inhibits the cysteine at the position 321. So these are the main target of different drugs. Up to date, five structurally related benzimidazole sulfoxide proton pump inhibitor have been approved to treat various gastric acid hypersecretory disorder, and these include omeprazole, isoprene, lansoprazole, pantoprazole, rabeprazole. So it is the general structure of proton pump inhibitor. So here there is a X, Y and Z, and here is a R. So based on this substituent, there are 5 proton pump inhibitor available in market: omeprazole, where X equal to CH3, Y equal to methoxy group, Z equal to CH3 group, R equal to methoxy group. Similarly, isomeprazole, same group is there, so it is an isomer. Omeprazole and esomeprazole is the isomer to each other, so same group is there. Lansoprazole, X equal to methyl, Y equal to CF3 CH2O, Z equal to hydrogen, R equal to hydrogen. In case of pantoprazole, methoxy, X equal to methoxy, Y equal to methoxy group, Z equal to hydrogen, R equal to CHF2O. Then rabeprazole, X equal to CH3, Y equal to CH3O, CH2 3 times O, Z and R equal to hydrogen. So these are the various proton pump inhibitor based on the substituent as a X, Y, Z and R. The rate of conversion of proton pump inhibitor to their active form sulfonamide is determined largely by the pK value of benzimidazole group. Omeprazole, lansoprazole and rabeprazole with higher pK value 0.6 to 0.79 undergo formation of sulfonamide faster than pantoprazole with pK value of 0.11. The proton pump inhibitor that inactivates cysteine 822, that is pantoprazole, tends to have longest duration of action because this cysteine residue is on the pump transport domain and resistant to reactivation and therefore require replacement of new pump protein. So depending upon the pK value of nitrogen, for example in case of omeprazole, lansoprazole and rabeprazole, the pK value of these drugs are higher, 0.6 to 0.9, therefore it undergo fast activation process as compared to pantoprazole with having pK value less, which undergo activation slowly. Then next point is the proton pump inhibitor which inactivates cysteine 822. So the drug which inhibits cysteine 822 like pantoprazole, so it shows longest duration of action because this cysteine at 822 is present on the transport domain and therefore it requires number of days to replace after damage. So it is about the proton pump inhibitor. So it is also important topic in exam. Number of time this question asked, that is short note on the H2 blocker and proton pump inhibitor. So it is about the antihistamine chapter. So in antihistamine, first we will see the classification. We were seeing the classification. Then first generation antihistamine, in that there is a SAR. Then second generation agent. Then mast cell stabilizer. Then H2 blocker. And then last one is proton pump inhibitor. So in next lecture, that is in lecture 4 video, we will cover structure of important antihistamine drug along with their IUPAC and uses. So if you like the video, then subscribe the channel. Thank you very much.